what I had already given you was the delta G naught values of each of the components. Okay, what you have to simply do was an addition and subtraction on both the sides. Okay, we had a similar slide in the in the lecture. So that was the idea. All you had to do was just keep adding the delta G naught values on both the sides. And if the ultimate value was negative, the reaction was spontaneous. If it was not, then it was a non-spontaneous reaction. Okay? Again, this is what I'll tell you about most of your exams, anywhere on this earth. Don't overthink the problem. Okay? And work with the information that you have. If I have to ask you to use this equation, then I will give you the value of the gas constant, and I will tell you the temperature to work with. Okay? So, and the other thing that most of you were going crazy about was the mixed inhibition graph. Again, it was the exact same graph from your slide. We have to work that mixed inhibition. And the very next question was, where would you expect the inhibitor from that graph to bind on the enzyme? Okay. Again, the information was on the same slide, and you just had to know that if it's a mixed inhibitor, then you would expect that inhibitor to bind either to the active site or to the enzyme substrate complex. Because it is something that is affecting both the Vmax and the besides that, most of the questions are pretty straightforward, and I hope all of you will do good. We'll see. We'll know it by next week. All right. So today we are looking at oligosaccharides or carbohydrates in general, not oligosaccharides. So this presentation is going to be divided into two parts. This is Thursday, today, and next Tuesday. Today we are going to just look at the basic chemical properties of carbohydrates, and next week we will finish the chapter by looking at oligosaccharides, that is disaccharides and the long chain carbohydrates, okay, starch, glycogen, and maybe their storage properties. So today we are just looking at the chemical structure and a few basic chemical properties. Okay, so why the name saccharides? Any idea? That would make everything a saccharide. Sugar. Saccharide is, or saccharum is the Latin for sugar. Okay, very simple. And as the name suggests, carbohydrates are the main source of carbon for most of the organisms that we know. Okay, there is hardly any organism on this earth that would not prefer carbohydrate as its primary source of nutrition, okay, or for cellular respiration. All the cellular respiration reactions that we know ultimately are burning down carbohydrates. If there is an organism that is exclusively given a diet which is not carbohydrate based, it's fat or proteins, you will find that the metabolic reactions first break down those components into carbohydrate molecules and then feed it into a cellular respiration cycle. Okay, that is why carbohydrates are so extremely important. Okay, so don't go by any diets that tell you to take low carbs. That's bullshit. I'm just kidding. The problem comes in when we are talking about processed carbohydrates. Okay, but carbohydrates are extremely important for any organism's survival. Okay, and again, so the basic formula, again, I'm not using slides today, but I will provide all this information in these slides to you. But I like to go with carbohydrates just to talk about, draw the structures so that you are aware of drawing them yourself, recognizing and identifying the individual carbon that we are talking about. Now, this is the general formula, CnH2n. Okay, n number of carbon, n number of hydrogen, water molecules. Water molecules, again, H2n means that each of the carbon or some of the carbons in the structure will have an H and a hydroxyl group on the both sides. So H2O. Okay. So CnH2n. And the simplest carbohydrates significance when it comes to metabolic properties, because the next part of the carbohydrate story that we talk about is glycolysis, which will start on Tuesday. Again, it will be split into two parts because it would form the basis of any kind of metabolic studies that we want to do. So you have the carbohydrate molecule either as an aldehyde or either as an aldehyde or as a ketone. Okay, this is a ketone group where you have the carbon in a double bond with an oxygen. Okay, and on both sides there are carbon units. Okay, that's a ketone. Okay, and if it is an aldehyde, this is the group. You have the carbon double bound to an oxygen and a single bond with a hydrogen. Okay, this is an aldehyde. Okay, now like I said, the simplest of all the carbohydrates that are metabolically relevant to us, okay, or where the whole carbohydrate chemistry starts, is with trioses or 3-carbon carbohydrates. Okay, this is glyceraldehyde. Okay, just recall the glycerol structure. Okay. It was this is your glycerol molecule. Okay, if you have one of the carbons, instead of carrying a hydroxyl group, it carries an aldehyde group. You call it glyceraldehyde. Okay, and glyceraldehyde, if it is a keto form or it carries a ketone group, it's called dihydroxy acetone. Okay. Now, again, start using, the information that is provided in terms of the names. Okay, because when we're talking about carbohydrates, we go into lipids, more into lipids. You should be able to decipher the molecular structure just by the name. Okay, so it says glyceraldehyde, very simple, that it is a glycerol molecule with an aldehyde group. Dihydroxy acetone, okay, or dihydroxy ketone, that means that it is a glyceraldehyde or a glycerol molecule with dihydroxy, with 2 hydroxy groups and a ketone group. Okay. So start using the names. Just by the names, you should be able to draw at least the most basic structure. Okay. Again, this is the beauty of organic chemistry. If you're just writing down the carbons and you are able to put the main functional group, the assumption is that whatever you have left blank is a hydrogen. So most of the time you're safe. Okay. So if I say draw dihydroxy acetone, if you draw something like this, the structure is absolutely correct. This and this are the same. Whatever you have not drawn, whatever valencies of carbon you are not showing, is automatically assumed to be a hydrogen. Okay. So just by the name, if you can draw something like this, you're safe. Okay. Now, something that we have seen before, or we have talked about before, is the chiral nature of amino acids. Okay. Anything that is bound to a carbon, okay, carbon has 4 valencies, okay, and the structures around the carbon are arranged in a 3-dimensional space. Okay. So there is a certain orientation in which you can look at the structure. Okay. The same structure can be oriented within the space differently. Okay. They are called mirror images, stereoisomers or isomers that we have talked about. Okay. Now this is what is the structure? Glyceraldehyde. Okay. You have the D form and the L form, again, dextro and levo, right and left. Okay. The only thing, or the only position where they differ from each other is the orientation of the OH. Okay, and I'm from the same thing.
You said the glyceraldehyde?
Yes.
Okay.
Okay. The right and the left orientation of the hydroxyl group on the second carbon. Okay? And if you have mirror images, okay, mirror images that are non-superimposable. Okay, so if you move this thing over here, try to superimpose this image onto this one, the hydroxyl will overlap with the hydrogen, and this hydroxyl will overlap with this hydrogen. So both these structures are non-superimposable, but just by looking at them, they are what you call as mirror images. Okay, so this is excuse me. This in carbohydrate chemistry is called an enantiomer. Okay, an enantiomer. What is an enantiomer? Something that is a mirror image of its structure, but is non-superimposable on its image. That is, again, it is a stereoisomer, but that stereoisomer is called an enantiomer. This is another very interesting structure we need to draw. So this is D erythros and D trios, 4 carbon units. Okay. Now, if you look at their structures, look at this hydroxyl group and this hydroxyl. Okay. On the second carbon, we can see that there is a difference in the orientation of the hydroxyl group. Now, again, so the carbon around which the functional groups are orienting themselves differently, what is that carbon called? Isomer? No, these structures are isomers of each other.
Diastereomers?
No. This carbon,
The alpha carbon?
is it the alpha carbon?
No. The beta carbon.
Beta?
That's what it's
Go ahead. Let's start talking in like, Greek now. Exactly. Remember, with respect to amino acids, we said that the amino acid structures are chiral. Okay? And remember, we were talking about only the main or the single carbon around which the functional group, the hydrogen, the amino group, and the carboxyl group were orienting themselves. Okay? This was your alpha carbon. Don't bring that terminology here. So, this single carbon around which the functional groups were orienting themselves, this carbon is called the chiral carbon or the chiral center. Okay? Now, looking at erythros and D-trios. Okay? These are non-superimposable images. Okay? But at the same time, they are not mirror images. Why are they not mirror images? It's the exact same chemical formula for erythros and trios. Okay? if I just give you the chemical formula based on this general formula, okay, you would not be able to differentiate between erythrols and thiols. Okay. This thing, or arranging the carbon molecules like this, is called a Fischer projection, named after Emil Fischer. We'll look into a few projection, diagrams besides Fischer projection. Now, again, I have not drawn a mirror plane between them. Okay, so you look at D erythrols and D thiols. At which point would you say that they are not mirror images? And again, in Fischer projection, whichever carbon is carrying the main functional group, either the aldehyde or the ketone, that is the first carbon. So second, third, and fourth. The third carbon, exactly. Okay. So the third carbon is where both the hydroxyl groups are towards the right. Okay. So when you have isomers like this, okay, which are non-superimposable but are not mirror images, okay, non-superimposable and not mirror images at the same time, they are called epimers. Okay. So when you have non-superimposable mirror images, such stereoisomers are called enantiomers. And when you have non-superimposable, non-mirror images, okay, these are called epimers. Keep these two definitions in mind. Do not confuse them. Again, I'll repeat again. Enantiomers are non-superimposable mirror images. Okay? And non-superimposable, non-mirror images are epimers. In both cases, you have the same molecular formula. Okay? Going into a bit of the chemistry of the whole thing. Again, everyone is clear about enantiomers and epimers at this point. Now let's look into the structure of glucose. First we'll draw the Fischer projection, and then we'll go into the Fischer projection again is the linear orientation. And then we'll go into the cyclic form, which is more common, when we are talking about the whole chemistry of the drug. This is your basic Fischer projection for a glucose molecule. Glucose is how many carbons? 6. It's a hexamer. Okay? And the main glucose that we talk about is an aldehyde. Okay? So you have the structure here. Now, how do we get a cyclic structure? Okay? Glucose, most of the time you'll find when you are, either ordering glucose or you're looking at the chemistry of glucose, you'll find that glucose is usually drawn like this. In its cyclic form. The idea is that the fifth carbon and the first carbon, okay? the aldehyde group, okay, and forms a cyclic structure. Okay, so the reaction that is taking place is you have the alcohol you have the hydroxyl group, okay? It binds to the aldehyde group, and what you end up with is with an hemiacetyl group. Okay, if it's a ketone instead of the aldehyde, it's called a hemiketyl group. Okay, so if I have drawn a structure like this and I ask you, what is this bond? This is a hemiacetyl bond. Okay, if it was a ketone to begin with, it will be a hemiketyl group. Okay, so if in this reaction, how do we get to this, or how we can draw this with all the hydrogens and hydroxyl groups arranged, is something like this. The first carbon and the fifth carbon are in a hemiacetyl linkage. Okay, you can see the oxygen being bound to both the first and the fifth carbon. Now, the orientation of the hydroxyl group at the first carbon becomes important when we are looking at the ring structure. Okay, if you see an orientation like this, it's the alpha. And if at the same time you see a structure like this, this is the beta. Okay? Again, these are all isomers of each other depending on the orientation of the hydroxyl group. On the first carbon, it will be either alpha glucose or beta glucose. Okay? So keep the orientation of the hydroxyl group in mind. If you see structures like this anywhere written either on the exam or wherever, and I ask you to give me the full name of this thing, then the alpha or the beta becomes very important. Okay? And there's we've already talked about the Fisher projection. When we are talking about the ring structures, there is another type of projection, and the ring structures, you'll find them usually written like this. Okay. These, the first three bonds that are towards you are Hayward's projection. Okay, very simple. If you're drawing just the, again, if you've just drawn this, then you don't really need to draw the hydroxyl groups around the thing. Okay, there is an assumption that yes, you know where the hydroxyl groups are. Okay. Usually you'll find this or something like this, just a bond going up.
Do you know right now where they are based on what you just drew?
No, no.
Okay.
So you don't even have to put the, you don't have to put anything, you just know that that's glucose?
Yeah.
Okay.
Okay. We need to look into the structure of fructose also before we move on. Now, the, what do you call it, there's another way that you write these structures. Okay, again, we are talking just about the stereochemistry or the orientation of the carbons. So when we are talking about the ring structure, one way that you can write them is like this. Okay. And I mean, whoever drew these out was like, you know, really paying attention to the names. This is called the chair. And this? Oh.
Yes, it's taking back.
Probably they got their kid to read the whole thing. Okay, so you can draw the glucose molecule or the ring structure, okay, either in the chair form or the bowl form. Okay, very simple. Why I'm giving you all of this information here might sound irrelevant, but the idea is that, you know, if you find them written like this, do not confuse yourself that, oh, this might be something different. No, it's just the glucose molecule. Again, we're just talking about the orientation of the molecule in space. Okay, and you can write them as a, again, as a Hayward projection, like this.
Does the boat represent the beta hydroxyl group?
Does the boat represent the beta hydroxyl group? No, not necessarily. Yeah. It's just, again, anytime we talk about structures, we're just looking at the energy stabilization, okay, in a given structure or in a given solution. You will find, because again, if I ask you that in a given solution, can you differentiate between the alpha and the beta form, or can you differentiate between the chair and the boat form? Can you differentiate between the alpha and the, what do you call it, even the keto and the aldehyde form? Not necessarily, just by looking at them. Okay? Where they start to make any differences when we talk about the stereochemistry with respect to the opacity of the structure, how they react with light, okay, or how they scatter the light around them. That gets into a bit of a detailed, you know, analysis of the whole solution, okay. But in any given solution, you'll find all these forms to be interchangeable, okay, depending on the whole energy environment of that solution. Okay. Now, pay attention to this hydroxyl group. Okay, so we'll come to that now. Now, reactions of monosaccharides. Okay. The most important thing that you need to keep in mind with respect to monosaccharides are that most of them are reducing in nature. Okay? What is a reducing agent?
Losing
It
electrons.
oxidizes.
Hmm?
Gain electrons.
It loses
Something
electrons.
that gains electrons, who said gains?
Loses electrons.
Something that loses electrons and gets
Oxidized.
oxidized. Okay? And so, what is an oxidizing agent?
Opposite.
All right. Most of the sugars, most of the aldoses,
Should ask last week.
sugars that carry the aldehyde group are reducing in nature. Okay? They act as reducing agents. Okay? Why do you think that diabetes is such a problem?
Too much sugar.
What is it that glucose is high glucose in the sugar doing?
Releasing insulin.
Hmm?
Releasing insulin.
Reducing insulin?
No, releasing too much insulin.
I'm talking about high glucose in the sugar.
Oh,
Okay. All right, let's not talk about diabetes in general. Let's talk about sugar, the problem that they call, oh, you have a sugar problem. You eat too much sugar. You have too much sugar in your bloodstream and in your urine. Okay. What is that situation? Why is it such a horrible thing when we do know that the whole system, all the cells are absolutely dependent on glucose? Why is having such, an important energy source in abundance in your bloodstream or within your cells such a problem?
Too much of a good thing.
Toxic, okay. Why is it toxic? High concentration. Most of the time it's not that high. It's just a little high and it starts creating problems.
Stored as fat or aspartate.
Okay. Again, think about the chemistry. Okay, not the actual thing itself. Why having higher amounts of glucose such a big problem?
Changes the pH.
Changes the pH. Not really.
Your body's
I just gave you one sentence about glucose being an aldose, and all aldoses are reducing agents. Reducing agents, you just told me, are going to gain electrons,
Lose electrons.
lose electrons, and react with all kinds of crap within the cell. So, what do you think is going to happen?
Too many electrons are lost.
Hmm?
Too many electrons are lost.
Too many electrons are being lost, and then what is happening to all those electrons?
All kinds of crazy stuff happen.
Exactly, all kinds of crazy things. No, that's exactly what is happening within the cell. Okay, all those extra electrons are just going to move around, start reacting with anything and everything that can accept those electrons. Okay, mainly the proteins. Okay, side chains of the proteins and the cell membrane, the lipid molecules, other sugar molecules that may be important structurally or may be important as functional groups. All those extra electrons are just going to start reacting. Okay, and that becomes a problem for the cell. When you have something that serves a certain purpose, so you have the proteins that are there to act as enzymes or act as structural proteins, act as channels, transporters, okay, whatever. And then you have the lipid molecules, okay, which are very important structurally, again, serve very important functional and signaling function. So you need those structures in their exact form for their various important functions. And now you have a higher concentration of something within your bloodstream, within the cell, which can start, donating electrons and start reacting with everything. That will basically, ultimately, kill all those molecules, okay? Or will hamper their functioning in a manner that is relevant to the cell. Okay? That is why having high sugar within your bloodstream, even though that sugar, glucose, is so important for your cell, for your body. Okay? That is why, the biggest reason when we look into the chemistry of the cell, what is the main transport sugar in plants? Is it glucose? No, I said transport.
Sucrose.
Sucrose. Sucrose is Glucose and fructose, it's a disaccharide. That's next to you. Okay? It's not glucose. Is there anything that exclusively stores glucose in the plant kingdom? There are a lot of, within the fruits, you will find a lot of fruits storing varying amounts of glucose. But is there like one that really stores a lot of glucose?
Chlorophyll?
Hmm?
Chlorophyll?
No, I'm talking about
Oh,
fruit.
oh,
Any idea? Corn?
Whatever malt liquor is called.
Okay, malt liquor is made up of what?
I said it's maltose, I don't know. Malt.
Kiwi? Grapes. Grain? Grain? Who said grain? Get up. Grains again. For next week, what? You'll be touched upon it. Yeah, Okay, grains are starches. Okay? Starch is nothing but a glucose chain which is thousands and thousands and thousands of molecules long. Okay? Okay, again, if we are on that topic, again, why I'm talking about all of this is because we'll keep coming back to the first carbon and the hydroxyl group here. Okay? Why do you think starch is being, why is such an elaborate structure made? When sucrose is non-reducing, we know that. Okay? This hydroxyl group is in bond with a fructose molecule. So why not just store everything as sucrose? Why get all the glucose molecules in like chains? And when I say chains, I mean chains that you can actually see with your naked eye. If you cut the grain, okay, and you have a decently powered lens, you can just see the starch rings very easily. Stable, sucrose is pretty stable. The reducing end has been taken care of. Sucrose is extremely stable. Do you see anything happening to the sucrose or to the sugar in your samples? It stays like that. Unless you're paying. And then, you have all kinds of shit throwing there. Sucrose is pretty stable is what I'm trying to tell you.
Something to do with efficiency possibly?
Efficiency in terms of?
Well, it's cheaper to store that way? So it just takes less energy to maintain it, or?
Storage. Okay. The simple idea is that you have a chain that can start to fold upon itself. Okay. So the chain is not just a chain like this. Okay. If you look at the starch grain, okay, it's a structure like this. And each of these lines is just the whole thing turning around itself. Okay. So what makes sense is, or for the plant, or for anything for that matter. You have a garden hose, just throw it out there. It's a problem it occupies a lot of space. Start folding it nicely and then fold it upon itself. You can have like a huge, 60, 80 meter hose in a very small area. Okay? And that is the idea behind making both glycogen, which is stored in the liver, and starch. Okay? If you look at the structure of starch and glycogen, they're extremely similar. There are just very, very few differences, which we'll go into next week. Okay? So the simplest idea is to put all the reducing ends of the glucose in a linkage, okay, so that nothing is being acted upon by the glucose, because the reducing ends are now in a link, in a huge link, and the whole structure is being folded upon itself to give you efficient storage. Okay? And that is why I asked you, what is the main transport sugar? Okay? The sugar is being made in the leaves and is required by different parts of the plants. Roots are not photosynthesizing, okay? And the flowers are not photosynthesizing. The fruits are most certainly not photosynthesizing, but they do require all the sugar to grow, okay? And that is why it is next to impossible to transport something like this. Okay? That is why it's a storage molecule. Okay? And the sugar that is being moved around in the floor is the sucrose, or the disaccharide sucrose. And it's not glucose because of the glucose, because of the reducing nature of glucose. And that is why you have the, the whole hoopla about antioxidants in grapes. Okay, grapes are not necessarily that bad, okay, till you are diabetic. The first thing your doctor will tell you is to stop eating grapes. Okay, and the main reason why grapes have such a high concentration of antioxidants is because their system somehow has evolved to carry more glucose and less sucrose. Okay, so the antioxidants in the red wine, which you might tell yourself at the end of the day, oh, these are antioxidants, because you're an alcoholic. No. Zerbitol, something that will save all of us. We'll talk about that sometime later. Okay, but all the antioxidants are there for the protection of the grapes themselves. Okay, now